This is lecture 10, and this lecture is about synthesis of commercial products by recombinant microorganisms. So we can introduce the catalytic activity uh, into a microorganisms by using genetic manipulation techniques, and thus make recombinant microorganisms. Then we can use these recombinant microorganisms to produce that product, okay, such as the restriction endonuclease, vitamins, amino acid, dyes, antibiotics, precursors of biopolymers, etc. Okay, so let's talk about in more details about this. First, restriction endonuclease. Production of restriction endonuclease using recombinant microorganisms. So restriction endonuclease, these are enzymes, and these are very important enzymes for research works, okay, especially the recombinant DNA technology. Restriction endonuclease, these enzymes are a very important key player in recombinant DNA technology, because in recombinant DNA technology what we do, we manipulate the genes, the DNA, okay, by inserting a certain portion or deleting a portion from the DNA, okay. So, and restriction endonucleases are required to do these things, okay. So basically, actually what restriction endonuclease do, they can cut the DNA, okay. So say for example we have a DNA and we want to insert a gene into that DNA. So first we need to cut that mother DNA, okay, and this cutting is, we use restriction endonuclease for doing this. So restriction endonuclease, they can cut DNA on specific places, okay. So restriction endonucleases, they don't randomly cut the DNA. Each, there are many different restriction endonucleases, and every of this enzyme, they have their specific regions in the DNA where they cut. So for example, E. coli is one restriction endonuclease enzyme, okay. It has a specific recognition site in the DNA. It cut only on that site, okay. Then another example, HIND3, it's another restriction endonuclease enzyme, okay. It's also have a specific recognition site in the DNA, okay. uh, Currently, there are more than 350 different RE, restriction endonuclease. Okay, RE are commercially available, and the total market size is $500 million per year, and this market is increasing. Okay, so from this number you can see, okay, it's a good number, right? $500 million, and as the market is increasing, so there is business. So anyway, the restriction enzymes, okay, which are used for research purposes, okay, in the rDNA technology. So these restriction enzymes are naturally produced by many different microorganisms, okay. So now if we want to use these microorganisms to produce the REs, the enzymes, in large amount, okay, so we have to use fermentation. But as different enzymes are produced by different microorganisms, so what that means, we have to design different fermentation protocol for each of these organisms. It will be a tedious work, right? So we can avoid this tedious work. How? We can clone the gene which encode the restriction endonuclease enzyme, and we can clone this gene into E. coli, okay. Then use that recombinant E. coli to produce that enzyme, okay. Okay, so okay, so why the microorganism they produce RE, okay. So basically it's a defense, it's a weapon for them, okay. It's a they the microorganisms, especially bacteria, okay, so they use this restriction endonuclease enzymes to protect themselves from invading foreign DNA or invading foreign, say for example bacteriophage, okay, or invading foreign particles, okay. So one thing is, so how the bacteria they protect themselves, as they can produce these enzymes. So whenever a invading organism can attack them, they use this enzyme to cut the DNA of that attacking organisms and kill it. Okay, so by this way they protect themselves. Now the fact is the bacteria who produce this enzyme, they themselves has DNA, right? So theoretically their enzyme should cut that means destroy their own DNA. Okay, so the bacteria they okay, it doesn't happen. Why? Because the bacteria they have a self-protective system. Okay, what is that protection system? The bacteria who produce restriction in the nucleus, they also have gene by which they can do methylation of the DNA, of their own DNA. Okay, so if a DNA is methylated, then this restriction enzyme they cannot recognize the DNA and cannot cut it. Okay, so now the fact is, say for example, a bacteria produce a restriction in the nucleus. So it is not necessary that their whole DNA have to be methylated. Okay, so every endonuclease, restriction enzyme, they recognize a specific DNA sequences. So the bacteria who produce the restriction enzyme, they only need to methylate that portion of the DNA which that restriction enzyme recognize. Okay, so bacteria protect themselves by their own enzyme by this way, through methylation of those recognition sequence. Okay, okay. Now the thing is, as I already said, that different restriction enzymes are produced by different microorganisms, and if we want to use those microorganisms to produce restriction enzyme, we have to develop a detailed fermentation protocol for each of those organisms. So in that case it will be tedious. So how we can avoid this tedious work? By using a different bacteria, say for example E. coli. Okay, so there are detailed fermentation protocol for E. coli. So now if we can change the E. coli by genetic manipulation, so that, and by doing this we can make recombinant E. coli. So that means if we can make a recombinant E. coli which will produce the restriction enzymes, then we can cultivate this E. coli by fermentation and produce lots of lots of these enzymes. Okay, so one example have been described here. Say for example P. stuarti. Okay, so P. stuarti, the full name of this bacteria is Providencia stuarti. Okay, so this bacteria it produce a restriction enzyme, PST1. Okay, this is not PSTL. Okay, this is PST1. So this bacteria Providencia stuarti, it produce this restriction enzyme. Okay, so now if we want to commercially produce PST1 restriction enzyme, we can do it by using recombinant E. coli. So what we need to do first, from the P. stuarti bacteria we have to identify the DNA that encode the PS2-1 enzyme. Okay, so if we can identify that DNA, then we have to insert the DNA into a plasmid. Okay, so for example PBR322, this is a plasmid. Okay, so we have to insert that DNA which encode the PST1 gene into this plasmid. Then we have to put this plasmid, cloned plasmid, into the E. coli. Okay, so in this photo, this is the DNA that encode the PST1 DNA isolated from the P. stuarti bacteria, and this is the plasmid which we will use, which is used as the vector. This DNA is inserted into the plasmid, so these are cloned plasmid. Okay, and then this plasmid are inserted into E. coli through transformation. So you can say these clone plasmid are transformed into the E. coli, and then through a screening procedure, this E. coli we have to select that E. coli which have this contain this plasmid. Okay, then This E. coli, as this E. coli have contained this cloned plasmid, they will produce the PST1 enzyme. Okay, okay. So now when we do this cloning, okay, so not only the DNA which encode the enzyme is inserted, along with this DNA some additional sequences are also need to be inserted, such as sequence for methylation, okay, methylation operon, okay, so and also the promoter sequence, okay, which is important for the expression of the PST1 gene. Okay, so this kind of sequences are also included in the cloned sequence. Okay, so this is the process. Okay, so by this way we can make a recombinant E. coli which carry a foreign DNA. Okay, so this foreign DNA, I mean, which foreign DNA is actually it's depend on us, right? So if we want to use, say for example, we want to use E. coli to produce Hint3. Say we want to use E. coli to produce Hint3 restriction enzyme. In that case, we have to insert the Hint3 gene into the E. coli. So by using plasmid, I mean by this way. Okay. Okay. So now one thing is, say for example, we know that P. Stuarti bacteria it produce the PST1 gene. Okay. So now how we can identify that which gene produce the PST1 enzyme? Okay. So basically in experimentally the basic is like this. So for example, first we know that PST2 produce the PST1 enzyme. So we have to extract the enzyme first. So enzyme is a protein. Okay. So from that enzyme uh, we have to get its amino acid sequence. Okay. So from the amino acid sequence we can get the DNA sequence. That means the sequence of the gene which produce the that restriction enzyme. Okay, so by this way we can actually get the DNA sequence from a protein sequence. Okay, so once we get the DNA sequence, that is the gene sequence, then we can use that gene sequence to make a recombinant E. coli, and then use that recombinant E. coli to produce that gene product. Okay, so it was about restriction enzyme. Then synthesis of vitamin. Okay, vitamin by recombinant microorganisms, and as example we will talk about ascorbic acid. So ascorbic acid is vitamin C. Okay, so vitamin C, I mean vitamin C is just its usual name, but its chemical name is ascorbic acid. So at present the majority of commercially manufactured vitamin C, which is L-ascorbic acid, is synthesized via a process described by Reichstein. Okay, and the raw material D-glucose is used to produce ascorbic acid. Okay, so the process which is described by Reichstein, it involves 6 chemical steps. 1, 2, 3. Okay, 6 chemical steps. Okay, and among these 6 chemical steps, only one steps is done by fermentation. Okay, so other steps are done by chemical uh, chemical process. Okay, so this is actually a expensive process to produce. I mean, although one step is done by fermentation, yet this is a expensive process. Okay, so these are the process. I mean the metabolic steps by which L-ascorbic acid, that is vitamin C, is produced from D-glucose. Okay, so D-glucose to D-sorbitol, D-sorbitol to L-lactose, L-lactose to 2-KLG, 2-KLG to 2-KLG sodium salt, then L-ascorbic acid. Okay. Okay. Now in these steps, only this step D-sorbitol to L-sorbose is done by fermentation process. Okay, so now as this is a costly process, okay, so if we can Why this is a costly process? Because only one step is done by fermentation, and all the other process is this fermentation process is a expensive fermentation process, because it needs lots of addition. Okay, very high level of addition is required, and as the other steps are done by chemical processes, so the total processes are costly. Okay, so now by using genetic engineering method, we can actually reduce the cost of uh, vitamin C production. How? Basically, by using genetic engineering process, recombinant microorganisms can be produced, which can ferment some of the step. Okay, thus replacing the required chemical steps, and it can reduce the production cost. Okay, so how we can do this? Okay, so it's already known that several microorganisms, okay, say for example some acetobacter, gluconobacter, okay, they can produce 2 kLg from glucose, okay, by a different pathway, not following this pathway. In this pathway, the 2 kLg is produced from D-glucose, okay, by this path, okay, from the D-glucose. But there are some microorganisms who can produce 2 kLg from D-glucose by a different pathway. So which pathway? Say for example, uh, Arwenia, this bacteria, Arwenia, okay, they can convert D-glucose into 2,5-DKG, okay, and another bacteria, Corynebacterium, they can then change this 2,5-DKG into 2 kLg, okay. So now, if we can use these microorganisms to produce 2 kLg, then we can replace all these steps, okay, and we can use the 2 kLg to produce the L-ascorbic acid. So now, the fact is, I mean, here are two different bacteria, right? So for example, Arwenia, it transforms D-glucose into 2,5-DKG, and Corynebacterium, it transforms 2,5-DKG into 2 kLg. So now if we want to produce 2 kLg by fermentation, then we have to use both these microorganisms. Okay, so theoretically it is possible to do co-fermentation. Okay, we can use both these bacterium at a time to produce 2 kLg, but co-cultivation or co-fermentation is a complex process, because most of the time these two bacteria, they may require different fermentation conditions. Okay, so now if their requirement for fermentation process is different, it will be very difficult to design a optimum fermentation process where both of these microorganisms will grow optimally. Okay, okay, so how we can solve this problem? Okay, so one approach to solve this problem can be by doing tandem fermentation. So what is tandem fermentation? Tandem fermentation is like, say, first we'll do only with one bacterium, such as Arwenia. So we'll do the fermentation only by using Arwenia to produce 2,5-DKG from D-glucose. Then we'll do another fermentation, second fermentation, a consecutive fermentation using the Corynebacterium to produce the 2 kLg. Okay. However, yet it remain complex. Okay, because in that case we have to use two different fermenter. Okay, and also the conditions, as the conditions will be different, maybe it will be required to change the chemical composition of the first fermented products. Okay, so that's why still it remains complex. So now, how we can solve this problem? Okay, so now by using recombinant DNA technology, so by using rDNA technology, if we can genetically manipulate one of these bacteria to do both of this work, then we can just do one fermentation to produce the 2 kLg. Okay, so now we have two bacteria. Which bacteria will be more feasible to change. Okay, now if we look closely into this metabolic pathways, so the transformation of D-glucose to 2,5-DKG, it's not a one-step process. Several processes, several steps are involved. Okay, so that means several different enzymes are involved in this process. Okay, so that means Arrowinea produce different enzymes to catalyze these different steps. So that means there are different genes in Arrowinea. Okay, but on the other hand, the Corynebacterium. Okay, so this Corynebacterium, they produce 2 kLg from 2,5-DKG in only a single step. That means only one enzyme. Okay, the name of this enzyme is 2,5-DKG reductase. Okay, so this enzyme convert the 2,5-DKG into 2 kLg. Okay, so that means there is only one gene. So that's why it's better. Like, if we want to what I wanted to say, if you want to change the Corynebacterium, then we have to isolate all the genes, okay, which are involved in this process from the Arrowinea bacteria, and then have to clone them into Corynebacterium. It will be a complex process. Rather, it's better if we can isolate the gene which produce 2,5-DKG reductase from the Corynebacterium, and then we can insert that gene into Arrowinea. Simple, right? Because there is only one gene. Okay, so exactly that is done. So the gene for the 2,5-DKG DKG reductase is isolated from Corynebacterium and then cloned it into Arrowinea, and thus we can the recombinant Arrowinea is produced, and then this recombinant Arrowinea, it alone can do all the processes. Okay, so then we can just the fermentation process for Arrowinea can be developed and use that and Arrowinea to produce 2 kLg by a single fermentation process. Okay, so if we can do use this recombinant Arrowinea commercially, then the production cost of uh, L the production cost of L-ascorbic acid, that is vitamin C, will be reduced greatly. Okay, so that's all for this part. In the next part, we'll learn about some other products which are produced by recombinant microorganisms. So thank you for your patience.